to get things started, Jim has told you a little bit of the sort of ADRC related background. And so, we do maintain this iPSC core at the ADRC, and we're trying to figure out what are some of the things that it can do and how can it support the community, but also how can we stimulate research? Are there research connections that can be made? And are there people who, might be interested or want to capitalize in these sorts of methods and techniques? So I'm going to talk a little bit about background, and pardon me if some of this background is super familiar to people. Just weren't sure that everybody knew what the ADRC is, what we do, and what we can do. And one reason to actually bring this up is that we are actively collecting skin biopsies, and we respond to the community in terms of very specific needs. So sometimes we think we know what people might use, but sometimes we get requests for things, and we can do our best to try and honor requests and come up with specific kinds of recruitments. So anyway, the ADRC is NIA funded. It used to be a P50 for the first 35 years. Now it's a P30. And what we do is we enroll people either who have normal cognition, mild cognitive impairment, Alzheimer's disease, some non-Alzheimer disorders, frontotemporal dementia, Lewy body dementia, and so forth. And we follow them through the clinical core. Our cohort size is usually about 450 to 500, and we're striving to have about 20% who are Latino. The cognitive normal elders, excuse the typo, should be age 65 to 100. Mild cognitive impairment, Alzheimer's. Within the Alzheimer's realm, we've been very interested in some of the variations that we see within Alzheimer's disease. And in the last few years, we've made an effort to enroll people who have younger onset of Alzheimer's, in particular below the age of 60. And sometimes these folks present not with memory loss, but with visual problems, which is called posterior cortical atrophy, or with language problems, which can present as aphasia. And then I mentioned frontotemporal dementia, which is itself an umbrella sort of diagnosis with people who have frontal lobe symptoms, disinhibition, and so forth, or language presentations or movement presentations, so progressive supranuclear palsy or PSP or corticobasal disease fit under the overall umbrella of FTD, and then Lewy body dementia. So the assessments that everybody undergoes.
Can I just quickly, just on top of mind when we talk about some of these subtypes, there's specifically in posterior cortical atrophy or regional sensitivity to neurodegenerative illness, we do have some philanthropy funds that are seeking to do pilot awards, and we'll stay tuned on that, but we'll be reaching out to the community about distributing some philanthropy money that would be specifically targeted toward regional atrophy like posterior cortical atrophy. I just thought I'd bring that up while we've got a good group here. Thanks.
Okay, so if someone enrolls in the ADRC, they undergo an annual clinical assessment, cognition, which includes detailed cognitive testing, medical and family history, and so forth. We collect biofluids. We have had a high rate of obtaining cerebrospinal fluid from newly enrolled people, and then we've been trying to repeat it at approximately 3 yearly intervals. And then we obtain blood for plasma and DNA every year. The DNA is used as a minimum for an ApoE genotype and for GWAS level DNA analysis. Through some large-scale national initiatives funded by NIA, there are whole exome sequence data on a subset of our cohort, and some are being subjected to an initiative for whole genome analysis. In CSF, we measure A-beta 42, tau, and p-tau 181 to help classify Alzheimer's disease biomarkers. On a subset of our cohort, we will include imaging. Some folks have had a research-grade brain MRI. Have had a tau PET scan. We follow people regularly and include attempts to try to get brain autopsies. And we've had an 80% autopsy rate over the past 35 years. These brains are maintained by a neuropathology core, and there's a standardized protocol. The brains are characterized, and we maintain fixed and frozen tissue. And so the bottom line is this is a resource for the community, and data and biospecimens are available. Just call us. So with regards to the iPS cell core, initially a collection of fibroblasts and iPS cells was started a few years ago by Larry Goldstein, and we've transitioned this collection and a number of other things we've been doing to a series of fibroblast cell lines and some iPS cell lines that are now maintained at the Salk Institute. We do skin biopsies for fibroblasts, and as I mentioned, some of these cell lines have been made into iPSC neurons. A couple of years ago, we participated in a SERM initiative where we actually had blood drawn and cells were transformed into progenitor cells, and those have been banked at Coriell, although I don't know that they've been very widely used. In terms of the kinds of things we have represented within the iPS cell core, we have many of the diverse conditions causing dementia, although it is mostly Alzheimer's-centric. And then we have people who were controls. And regarding controls, we have not sort of gone out of our way to biopsy people in their 40s or 50s or 60s, because it's really not clear if someone is doing cognitively well at age 50, whether they still will be the same way at age 80. So we've collected controls from people who've kind of stayed controls, and many of the controls have been in their 80s when they've been biopsied, and we've continued to follow them. There are some monogenic disease cell lines, so some presenilin 1 mutations, an APP mutation, and an APP duplication. And Jeffrey Jones is the acting director of the iPSC core and can help people with requests. So this is a snapshot of the cell lines. These are fibroblast lines. As I say, some of them have been made into iPSC lines, iPSC neuron lines. So 24 normal controls, age 70 or older, 20 late onset ADs divided by ApoE genotype, 20 young onset Alzheimer's disease. Many of these are non-familial. There are some really quirky things about young onset Alzheimer's that deserve further exploration. 4 lines from monogenic familial Alzheimer's disease. There may be some more that a couple of individual investigators have gotten from other sources. 5 cases of DLB, and 1 case that we thought was Alzheimer's but turned out to have this interesting entity called late, which is TDP43 pathology developing later in life. DLB and late are hot topics in the neuropathology and research community, and I think there are lots of opportunities to try and figure out how iPSCs could play into trying to discover more about mechanisms of both of those 2 disorders. So some other resources. So, we have our collection. UC Irvine ADRC also has an iPS cell core and are certainly more than willing to share cell lines. And there are a number of other ADRCs that also have developed iPSC cores. Regarding tau, the Rainwater Foundation was established a few years ago, and they've put together a number of different academic sites to try and do research into tau. And they've collected a fairly large series of skin biopsies from people who have tau mutations and also cases of sporadic PSP or corticobasal syndrome. And these are housed at Washington University. For Parkinson's disease, there are skin biopsies available through the Parkinson's disease biomarker program and affiliated resources run by NINDS. And then very recently, NIA made an announcement that they were using some iPS cell lines that they were going to make into neurons and astrocytes, and they were going to knock in a host of different neurodegeneration mutations, several APP mutations, PS1, PS2, tau, TDP43, alpha-synuclein, and they were going to try and also knock in a few other polymorphism type variants that are associated with Alzheimer's and other disorders. They're claiming to then be able to grow these neurons, astrocytes, and so forth, and perform RNA-seq, proteomics, and attack-seq on all of these cell lines. And that is going to be an informatics resource for the community. So these are some of the resources that are available. And I took a stab at some of the research questions that may be relevant to Alzheimer's and related disorders that iPSC resources can support. So the thing that's been done the most commonly is to look at monogenic routes to disease. So for example, presenilin 1, presenilin 2, APP mutations, and also I omitted Down syndrome in this list. And certainly for mutations, ideally one should be able to then compare with isogenic control lines and look at whatever phenotype one is interested in doing. Other genetic risk factors, there's a fair amount of research people have done with iPSCs in defined ApoE backgrounds, so E2, E3, or E4, and either these were done with skin biopsies or blood drawn from people who were carriers. In some centers, E2, E3, or E4 was knocked in against a neutral background in the same cell lines. TREM2 is a hot topic, in particular in relation to microglia. SOL1 is being looked at in relation to axonal transport and trafficking, and there are some mutations in SOL1 associated with young onset Alzheimer's disease. And then there might be an opportunity to try and dig into mechanisms of polygenic risk with some of the large international gene sequencing efforts. There are now probably over 80 different risk alleles associated with Alzheimer's disease and polygenic risk scores that have been developed. Regarding sporadic Alzheimer's disease, there are questions about how can one detect or study what the effects of age or sex might be. And I know Rusty will talk more about this, that in addition to looking at traditional iPSC types of methods, which rejuvenate cell lines, one can make directly induced neurons. Then there are a host of neurodegenerative disorders which have a complex etiology. Maybe there's some genetics at stake, maybe some aging, maybe some other factors, and there are no really good animal models. And so in the alpha-synuclein realm, dementia with Lewy bodies would fit into this category. Actually, so would Parkinson's to some extent, and multi-system atrophy as well. I mentioned this entity of LATE, which is late life TDP43 related pathology, which is an important cause of dementia in people in their 80s and older. But then TDP is also associated with frontotemporal dementia. And then PSP and corticobasal disease are very poorly understood, and it's conceivable that iPSC resources could be used to study those disorders. And then drug discovery, validation of mechanisms, toxicity, and going way beyond deciding let's make neurons in a dish or let's make astrocytes. It's possible to look at much more complex cell organization, organoids, 3D culture, transplants into animals, and so forth. So a host of different interesting opportunities. And so for tonight, we've rounded up some researchers in the community who are going to tell us more about this. I was going to finish with, I did a literature search looking at publications that actually came out of skin biopsies or resources where we had actually collected the materials at UCSD. There are a few more beyond this, but this is sampling over kind of the last decade, where Larry Goldstein got a number of things going with some of these early papers, and then other people have joined in. And so clearly there's a history here, and we would like to be supportive and try to keep adding to this.